Okay, so I want to welcome everybody to the another ESEP webinar, and today's speaker is Professor Maggie Lau. And since I don't have her bio in front of me, I'll let Maggie introduce herself.
Sure. Hi, good morning, good afternoon, good night. Thanks for being here. This is actually my very first webinar during this COVID time. And here you can see the campus where I'm located is in Sanya, but I'm physically in Hong Kong, my hometown. I can explain that later. So let me switch it to the laser pointer. So I started my job at the Institute of Deep Sea Science and Engineering in Sanya in the fall of 2018. I'm one of the main PIs in the Laboratory of Extraterrestrial Ocean System. I call it LEOS. So although I have a Twitter account, but I don't really tweet a lot, so I'm sorry if people feel like I'm not responsive. And so since the very beginning, when I decided to do a grad program, I fall in love very much on extreme ecosystems. I find it very fascinating, and going from the perspective of microbial ecology to environmental microbiology, and then later on geobiology, and now getting into astrobiology. So I get my bachelor degree, PhD degree, a postdoc, in the University of Hong Kong, and at that time, Professor Stephen Pointing was in Hong Kong, and now he's in Singapore at Yale NYU. And I consider that having just one advisor, I should go aboard and broaden my horizon, and before I start my own lab. And so I have the privilege to join Professor Toes Onstock at Princeton University in 2011. And I love the place, the group, and a lot of interesting projects going on there. And so I thought I would be there for just 1 year, and I ended up being there almost 8 years. And it was very rewarding. And thanks to, we still work together now. And then, as I said, I moved to Hainan in 2018. So the talk today is about astrobiology, of course, but through the lens of geomicrobiology, where I am more strong in my research field. And so what's extreme ecosystem? So extreme is only to us according to the human standard, but for those who can live there, it could be their optimal condition. So a quick quiz. Do you know the limits of the human body? Okay, so here's something I found online. So about the limits on body heat, altitude, and the depth and oxygen level. So correspondingly, you can also see that the current known high temperature or the most thermophilic organisms in archaea that can grow at 121 degrees C. About low temperature. So I'm not sure whether you're aware of it, but it has been found that this archaea is metabolically active even at minus 25 degrees C. About low pressure. So Andrew Sugar conducted a study and find that some bacteria can grow even at 7 millibars. So the challenge is that the lower partial pressure makes the substrate concentration so diluted. And then about high pressure, deep sea environments, even deep subsurface, they experience the hydrostatic pressure to a very high level, but the recent study also shows that you can find lives even down to 10 kilometer deep, and subject to 1.8 gigapascal, they still survive. And so this is just a short list. Extreme conditions include UV intensity, salinity, pH, and also other kinds of extremes. And so the talk today, I mainly draw some examples from my previous work and try to establish or review the links to astrobiology if it was not clear to you. And then at the end of the talk, I will introduce a little bit more about the Institute and also what I see about the astrobiology in China. So I work with a lot of extreme ecosystem, which I found very fascinating to me, including atmosphere, geothermal spring, deep subsurface, permafrost affected areas, and the desert area. And so each of them may have one or more extreme conditions. For example, high UV or high radiation, low temperature, a combination of light independence or low carbon input. And so all these extreme environments have relevance to potential life beyond Earth. And in the heat of discussion will be Mars and also the icy worlds. And the picture is not included here, but early Earth environment also resemble a lot to these extreme ecosystems. And so the first environment, as I promised in the abstract, we're going to 4 different environments. So first is geothermal springs. So here is a map of hot springs in China. And it's actually a paper map I scanned page by page when I was doing my PhD. And nowadays, you guys are more privileged, you have a lot of digital map to work with. Anyway, it's only to show that where the hot spring I work with is where the yellow circle here show is at Tibet. So I got to go to visit this hot spring in Tibet at around 5,050 meters in altitude. And so the hot spring is interesting. It is a neutral, almost neutral hot spring with a silica deposits around the rim here. And then the temperature is actually not very high, so only 80 degrees C compared to hydrothermal vents in the deep sea. So as it flows, it actually joins a river nearby. It drops down to about 30 degrees C. So the ambient temperature at this time is around, I think, 50 degrees C, or at night, it could be 12. But the water keeps flowing and it's warm. So you can see that the microbial mat is really thick. The scale bar here is about 1 centimeter. So one of the experiments I did was, okay, so we want to understand how disturbance may affect the ecosystem and how long does it take for the mat to grow back. And so I did a very short time experiment actually. Just grab some glass slides from the lab and then with the clamp secure them at each of the spot indicated by the letters here along the thermal gradient. And then we left and then do other experiments, drive off to other sites. And about after 5 days, we come back and check on the slide. And it was really a primitive idea when I first set up this experiment. And it was surprising to me after 5 days, there was something. And so I took them to the lab and then used SEM to analyze them, the slides from different temperature. I quantified the cells based on the morphology, like the rod shape or the filament. And in terms of rock shape or coccoid shape, I also categorize them by size as well. So the limitation of the SEM is you won't be able to get a precise identification. But through the SEM, I was able to tell that in the 5 days incubation, we see amorphic silica formation on the slide. They appear as a laminate or spheres, or we even actually see them deposit on the cells, or in other words, the cells is a nuclear for the minerals to deposit. And so the main point here is to tell you that in the hot spring, as the silica deposit, the silica actually deposit as the biofilm grows. So what does it mean? So the thermophilic organisms, they grow in an environment in general seems to be barren of life, but the hot springs serve as an oasis. They actually grow fast and they have a chance to be fossilized. And that means they can be preserved. Maybe their organics will be degraded over time, but their behavior, for example, their burrowing, would be documented in this mat. And so it has been shown in the deep subsurface, in the deep sea as well. So Professor Peng from our institute published a paper, their group published a paper in 2018. They showed that the burrow actually can go up to a size like 10, almost 10 micron in diameter, indicating that it's not just microorganisms, but also macrofauna would result in this kind of mineral deposition. So what does that mean? That means maybe in some place on Earth, or maybe in Mars. Mars have actually been shown that there's a possibility hot springs may have been present in the past. And also recent data shows some hydrothermal mineral deposits near the caldera. And then of course, in the early Earth, people talks about the hot springs could be one of the way to promote the emergence of life. And so these behavior and their biomineralization capability provide us one way to search for life or to understand the life in the past. Moving to hyper-arid regions, here shows a map of what we define as hyper-arid or arid regions. It actually defined based on the precipitation to the potential transpiration ratio. And so the study sites that we look at is also in Tibet. We're indicated by the bubble here. It's classified as polar desert and also with the very low precipitation, annual precipitation. And this site and overviews as Carmen here, and then we chip off some rocks from the hillside here, but also lying around are what geologists call flows. They are small pebbles or rocks, and they are sandstones. And with a hammer you crack open, you will see a very nice band of green, usually green and sometimes white or pink. So they are actually live. When you look under the SEM, you will see that they're actually a big collection of colloid cells as shown here. So in Tibet, at such high altitude, we actually measured the photosynthetically active radiation, the UV level as well. On the noonday, so this is the level as shown on here. So to give you a reference, at the sea level, the UVB level is 5 times lower than that in Tibet. And so we have done a test to see why or at what depth, why these organisms appear at such depth. So we measure the distance of the colonies or the colonized band from the surface of the rock. And then when we go back to the lab, yes, it is in the lab, not in Tibet. We actually chip the rock, this part, to a similar thickness. And then on the noonday, we measure the transmittance through the rock chip. So this is the result. We have different type of rocks, and here the radiation type. On here, it indicates the transmittance in percentage. So that means about 5% of light get through the rock chip, and here's like 2%. So on average, more than 95% of the radiation, regardless is photosynthetically active radiation or UV level, were filtered out. And in general, they colonize at a depth that is between 1 to 4 centimeters. And so to further understand what they are, and so we use a technique that characterizes the 16S for bacteria and 18S for bacteria or archaea, 18S for eukaryotes. So 16S has conservative region that's shown in green here, and also 9 variable region, which helps us to differentiate species two species. And so what we do is to target a short stretch of this ribosomal RNA gene and quantify them using quantitative qPCR. Quantitative PCR, or we call it qPCR. So the main point I want to say is there are about 10 to the power 6 of a number of copy genes here. So usually one cell has one 16S gene copy. E. coli sometimes will have up to 14, but in general, it indicates that there are about 10,000 to 1 million cells over 1 by 1 cm square area. So if you go out to the beach and swim, if you scoop the water out, then the seawater on the surface level has about 10 to the 5, 10 to the power 5 number of cells. So that's a lot of cells in that green band. So what are they? So you can tell it's like before the next generation sequencing era. So we use a technique called DGGE that helps to separate the 16S from a mixed community. So they will appear as discrete bands on a gel, as shown here. And then each band will excise or cut out and then take for sequencing. And so the diversity is actually quite high, ranging from proteobacteria to cyanobacteria. We have also fungi to form lichen and archaea as well. So further, we because it's a green band, it indicates that photosynthetic organisms could be the base or the dominant organisms in this community. And so we constructed a phylogeny tree for cyanobacteria. So the green arrow indicates the organisms that we obtained in this study. And then the green box indicates a lot of them come from a genus called Chrocococcidiopsis. And we actually did a follow-up study on the Chrocococcidiopsis because it is well known to be found in desert area. And so through the collaboration with NASA, and also we sample some of the samples by ourselves. And so we collected samples from, we define it as hot deserts, indicated by the beige box, and also cold desert, indicated by the turquoise box. From there, we amplify not only just the 16S part, as indicated from the in the previous slides, but we actually obtain a much longer sequence extending into the early parts of the 23S. And so what are we trying to achieve here is we have some hypotheses for how to explain possible global distribution of Chrocococcidiopsis. One possibility is because the samples come from all different regions, maybe they just have a stochastic distribution, have mixed each clay. So the closer are the box, that means they are more related. So they appear as mixed geographic regions or mixed environments. Or they're grouped by they're grouped by geographic regions or environments separately. And so as I get you ready, here's the result. So what we see is of all the strains that we obtained from all the different locations, we find out that the strains from the hot deserts and the cold deserts actually separate very early on in the history. And so how we actually constructed this temporal phylogeny is at that time, by then, when we were doing this analysis, there were 2 fossils retrieved from, one of them is from Australia, the other one, sorry, I forgot. And so when we performed this phylogenetic analysis, we used those 2 fossil records to as an anchor for establishing the molecular clock for the whole cyanobacteria tree. And then from there, we use that as an anchor for constructing the relationship between the Chrocococcidiopsis strains. And so you can see the common ancestor when the hot types and the cold types separate, dated back to 2500, which actually predates the contemporary environmental condition. the other way around. So this actually resembles to the third hypothesis. So the environmental condition actually is a very important determining factor for these organisms. So these organisms actually has been living on the earth for so long. So, and they live in the porous space in the rock. And they obtain their food by photosynthesis. So what does that mean? So this is related to some people, some scientists talks about, maybe you have heard the term before, panspermia. That means where lives come from. Would that be some of the organics or cells coming from outer space when they hit the earth? They inoculated the earth with this organisms or organisms similar to Chrocopsidiopsis, and then a given time they evolved and then become human or diversifying. And so although this picture shows that it is the inoculation into the earth, but it also means that implied that interplanetary transfer of biological material may be feasible, and Chrocopsidiopsis with its characteristics listed here would be a very enticing candidate to further study this possibility. And so I'm not saying that because we discovered this, so people talks about that. And long ago, long before we conducted our own study back in 2007, this may not be the first study, or maybe, sorry. So Professor Coquel in UK, they actually conducted a study. I would say that it's a very bold move. So they cut the nice to 1 cm thick with a 20% porosity, and then they immerse that into a culture solution with Chrocopsidiopsis. And then afterward, they put them onto the external surface of the spacecraft capsule, which would be launched by EAS. So they launch and then let it sit on the orbit of the Earth for 16 days, and then they orbited and then actually retrieve the samples from here. At the time when they retrieved, they realized that, oh my God, the samples were ablated and melted. And so when you look closely, they actually form a glassy fusion crust. And looking at under the SEM, you can identify the compositions mainly composed of feldspar and quartz. So regarding the question about panspermia, what's the answer? Unfortunately, the experiments find that there's no cells left, and not even DNA. But they use this melted or glassy crust. They re-emerge them into the culture and realize that maybe this process entering into the earth changed the structure of changed the structure of this rock chip, and which somehow enhance the cells to grow faster, and they colonize to a deeper level. And so compared to, so these are the Chrocopsidiopsis on the chips that have not been launched as a negative control. And so although this experiments indicates that everything occurred once entered into the Earth's surface, but that has not stopped people from continuing the work to study this behavior or the potential survival of organisms under high radiation and also impact shock. So the goal of my talk is not to give you every detail of what's going on in astrobiology, but to bring into light that you may search more if this interests you. Okay, the next situation is permafrost affected areas. And so there are lots of interest in the permafrost area because of the carbon cycle and also global warming. Let me explain why. Because in winter, they are under the snow and frozen. But then as the summer comes, they become warm and the bacteria would be active. And as the temperature get warmer because of global warming, then that means more cells will be active and the cells will be more active. And the active layer here are the layer where it will thaw during the summertime, but the soil column below that remain frozen. That's how permafrost being defined, is they remain frozen for 2 consecutive years. And so then the problem is, as the carbon, the organic carbon stored in the permafrost may not become available for the active microorganisms, and then what would happen? So it would be mineralized into CO2 or methane. So that would further intense climate or warming situation. And so our study site is at Mars Station. It's actually McGill Arctic Station. McGill Arctic Research Station, Mars, is right here. And so we collected some cores while it is still frozen. I was not there, unfortunately. I wish I was. So the cores here we extracted go all the way down to almost 1 meter. And you can see the TOC level is a little bit low. It's actually quite low compared to other permafrost people have been studying. It's around 5% fluctuating in the upper level, but then it dropped quickly and remained at 1%. The soil is relatively acidic, around 5 or 5.5. And so what we did is, with all the cores, we have 21 of them, we separated them into 4 different treatments. One, control, saturated means we put artificial water to mimic potential saturated condition. And also drained, which if they are on a slope, then the hydrological condition will allow the water to escape. So there's no water accumulation. And then the edge, where the ventilation would be better, and usually you will see less grassy material or plants growing on the edge. So we want to see the effects of them. Although in my talk, I won't go into detail, but you can go and check out Brandon's paper. And we extract the headspace. The headspace, we use an artificial air, which compose basically the same atmospheric composition, O2, methane, CO2, and nitrogen. We take out the gas for GC analysis, gas chromatography, to monitor the composition. At the same time, we have thermocouple to monitor the temperature of the core, and we extract also the pore water for pore water chemistry, and the soil for both organic composition, characterization, as well as where I come into place, the microbiology part. Not only me, I work with a team. And so for those who are not biologists, you know, the cells function starting with DNA, and as they transcribe, they produce RNA, usually it's a messenger RNA, and also the ribosomal RNA. And then they further being translated into a protein, which will function, has enzymatic functions. So the way to categorize the total DNA, total RNA, and total protein are called metagenomics, metatranscriptomics, and metaproteomics. I'm not going to spec out all the details of how we perform, but the results here is, interestingly, we find out a gene in both the RNA fraction, DNA fraction, and protein. So go back a little bit. So detecting DNA only indicates the potential of a certain function, but we need to further evidence from the RNA portion and also protein portion to indicate that this function is actually actively going on in the sample. And so here, putting them together, indicating this gene is actually a marker gene for aerobic methane oxidation. So we find out that it is closely related to a type of organisms called upland soil cluster alpha, USC alpha. So what are they? They are actually found in both in desert, in temperate region, in the soil. And they are known to oxidize methane even at the atmospheric concentration, which is very low. At this time, I think it's around 1.86 parts per million in volume. So what's special about these things is we're the first time to report the active organisms with evidence that they occur in the Arctic Circle. And so what does that mean? Before our study, actually we questioned ourselves, is it sure we actually have this organism? Because all the study at that time is about methane being emitted from permafrost, or when permafrost thaw. Here I can show you the places where have been reported as a methane source in the flux. And so complementing the microbiology data, we also have the gas sampled out, and here is a relationship of methane flux in our experiment and also in the field as a function of temperature. Here, the higher, the more positive means the higher methane flux. Here, flux means uptick. And you can see that as the temperature increased towards the right, the methane flux increase as well. And here the blue dots are actually a measurement done on the field, not just in our core experiment here. And so what it shows is atmospheric methane flux actually increases with increasing temperature. And they actually change very quickly between this time. And as it goes above 5.6 degrees C, it actually doesn't change much. So the temperature change over here has a more significant effect. And so we're the only study. That seems odd to me. And so go dig deeper in the literature, and you actually find more studies that actually have been reported that the site is a net sink of methane. And so go even deeper in the literature, you can find that the literature or the sites that have been reported as the methane source, they actually, in the table, in the paper, they have negative values. That means in their data, they have seen methane sink, but no one talks about it. And so we estimate in this area, in the permafrost affected area, 87% of the aerial coverage has low carbon content as our sites, the yellow dot. So we postulate that we have the hypothesis that these low carbon permafrost sites could play a big role in absorbing the methane. So I won't go into detail, but we recently, early this year, have the regional modeling paper published, which do show that the atmospheric methane uptake, when considering these methanotrophs, would take up a significant amount of methane produced by methanogen. So you can check that out. So back to today's talk about astrobiology. So knowing that these methane oxidizing bacteria can take up methane at such a low concentration, and they live in area where there's the water level is low, then what does that mean to astrobiology? Some of them may already have it in mind. Martian atmosphere. So the main component, the two should be subscribed. So methane actually have been reported repeatedly, like a cycle. Although we don't know how to explain this cyclic fluctuations of methane on the Mars, but there's stories showing the biotic and also abiotic reactions or a pathway that produce methane from the subsurface. Maybe microbes involved, producing, degassing, and then methane is known to even on Earth, 90% of the methane in the atmosphere are actually destroyed through photochemistry, not by microbes. And so these things also happen on Mars. How about would microbes also play a role on Mars in absorbing or taking up methane? I don't know whether people are looking at this. I wrote a proposal to NASA, but it was not funded. It could be my proposal was not written very well. But anyway, I still think that potentially the fluctuation of methane on Mars could be could involve life. All right, and I realized that actually last year, November last year, there was data showing that not only there's methane cycle going on Mars, there's also oxygen. Unlike other gas, so CO2 back here. So CO2 is the dominant gas. So they found that nitrogen, argon, they follow the fluctuation of CO2. So suggesting that it's more a chemical process, or as the temperature change, so the contraction of the gas change the concentration, overall concentration. However, the oxygen doesn't follow. Interestingly, the oxygen fluctuation tracks better the methane fluctuation. What does that mean? So the authors, they said they don't know, and explanations are wanted. Maybe with your expertise, you're able to help solve this puzzle. So lastly, talks about deep subsurface. So as we go into the Earth, there are organisms, but the cell concentration actually decrease with depth. So as we go down, actually the temperature increase, it gets warmer because it gets closer to the core. The pressure also increase as well. As you go down, you also receive more input from, we call it geogas, methane, hydrogen. They are important energy source for chemoautotroph, for organisms that live on chemical energy. So they are produced deep down through geological processes. And also one thing I need to bring out very important is in the deep subsurface, the influence of light is very little as you go deeper. And also the photosynthetic, for the products, organic products generated by photosynthesis, as they percolate down, they will be used up by the organisms at their cellular surface. That means as you go deeper, organic input also gets become little. And so I'm going to introduce a term called subsurface lithoautotrophic microbial ecosystem. So we call it SLIME. It is a system we consider that the subsurface do not live on anything related to a light source, including the organics. And so it is known that there are methanogens, new organisms, and also acetogens. They synthesize methane, they reduce sulfates, also synthesize acetates. So these are known organisms from early on when studying the deep biosphere in the continental subsurface. So what do you need to remember is hydrogen has been known as an important energy source, and hydrogen support all these organisms. Let me check the time. Good. And so one of the paper published by thesis group and co-workers is they found that a sulfate reducing bacteria, which has not been isolated then, but it has been isolated recently, interestingly, not from deep subsurface, but from Siberia, shallower subsurface, not in the deep subsurface. And so this is very basic graph, but I'm not going to tell you or explain anything on the cell here, but I want to tell you how these organisms live in the deep subsurface. So in the deep subsurface, there are different minerals and rocks, including uranium-containing rocks. So the radiation from the rocks actually splits the water that provides hydrogen. As I mentioned before, hydrogen is an energy source. And also the radiation generates oxidant. The oxidant will react with rocks to produce different nutrients. For example, carbonate and also ammonia, which means it has energy from hydrogen, has food or nutrients. The carbon source and nitrogen source, the main ingredients for life, are available through radiation in subsurface. So that's a very unique example in the deep subsurface. And so I was part of a study that involved going into a gold mine in South Africa in Beatrix. So they're very nice to us. They keep the borehole for us for 3 visits over the past few years. And it's located at 1.3 kilometers below the land surface. So you're familiar with the words now, the metatranscriptomics, metaproteomics, including with, and also with a geochemistry characterization as well. What is that? And so our interest is to find out what is alive, not just what's there. And so we focus on the RNA and the protein fraction of the biomass. And what we find is actually a very, very diverse community that are active at the 1.3 kilometer deep location. So 33 phyla actually are located, so including archaea and some eukarya as well. But the main thing that I want to indicate is the dominant part is proteobacteria, and which is affiliated to those who are capable of oxidizing sulfur. And so, the oxidation require require nitrate as the electron acceptor, which previously was not recognized. And then the other thing we wanted to find out is how do they live? And so it is well known that AME is the anaerobic methanotroph. It's part of the archaea family domain here. And so they form a tight relationship with SRB, which is the sulfur-reducing bacteria, by exchanging electron and potentially by exchanging sulfur species as well. And as we know that there are methanogens, and so methanogen means they are the partners that produce the methane for the anaerobic methanotroph, the AME. So for the sulfur-reducing bacteria, where do they get sulfates? As I mentioned, there's a dominance of sulfur-oxidizing bacteria. So they may also form a tight metabolic relationship. And so for each of the biochemical pathway, let's see how much energy can they get out of at this environment, given the concentration of these salts. So the number shown here is the free energy flux available through this biochemistry pathway. So these numbers may not mean much to you at this moment. So let me tell you what's the minimal energy requirement for sustaining a cell. It's 10 to the power minus 20. So these organisms, we consider that they are living on the edge. That means they don't get much energy. And so we consider having a tight metabolic relationship then through the frequent exchange of metabolisms. Basically what SOB doesn't want become the desired food source for the SRB. And so on, and same for the AME and the methanogen couples. And so the main message here is to tell you that in the deep subsurface, for them to survive, one of the way is to establish a syntrophic or mutualistic partnership with each other. So it stabilizes the ecosystem, the biological component. Okay, back to the astrobiology. Seems a bit diverted, right? But not really. So the subsurface is autotroph. They live independently from light and also photosynthetically derived organics. They could be self-sufficient, or they form a very strong independent network. So what's the implication? We consider that subsurface could be the most probable habitable environment on Mars. Given on Mars there's a lot of high energy particles bombarding the surface. And so different high energy particle actually penetrates the surface at different level. And so recent study actually try to model as the energy particle enter through the atmosphere, they will hit another particle, have a second generation of high energy particles as they propagate down to the subsurface. So the study was trying to find out where at the subsurface would be a okay depth for life or a human being to be there. So this is the result. The table actually shows with the different types of rocks, the shielding effect would be different. And here shows the depth where you may receive different dosage of energy dissipated from the high energy particle. Okay, so how to interpret this? So give you a reference. I gathered from the World Nuclear Association. A typical background for a human being to experience in a year is 2.4 milli Soviet. And at 100 m's Soviet, it's already a level risky for getting cancer. And so you can see that at around 100, so the shielding depth is around 200 or up to 200 cm. So it's actually quite shallow. For the deep subsurface organisms that I just talked about, it actually live a kilometer deep. And so it's possible that they are there. And also thinking about the surface temperature on Mars is 100 to 200 K. And so at this, at the 200 cm depth, so the temperature may not be good for the organisms, but as they go deeper, it get warmer. And so, yeah, so deep subsurface organisms is an interesting subject to study regarding to their relationship to astrobiology. So that's all about the science part. Now, I quickly go through the remaining few slides. So IDSSE is not as young as the new center led by Sid in India. So the whole concept of having a deep sea science and engineering institute was conceived and approved in 2011. So it was officially started operating at the campus. It's officially opened in 2016. And then in 2016, the research vessel has gone out to Mariana Trench. And a manned submersible also joined the later cruise as well. So in the past few years, actually, 探索1号 has already, I think currently is on the 16th cruise. And more excitingly, the second vessel has already set off, actually right now, set off for testing. And why we have a second research vessels is because the manned submersible 深海勇士 only goes down to 4,500 meters. And so to get to the Mariana Trench, previously we used landers for doing all the automated sampling and measurements. So with the new manned submersible, which is anticipated to be ready for sea trial by the end of this year, it actually go all the way down to the 10,000 meter deep. And so with this larger submersible, so we require a second vessels for launching and also recollection. And so in our institute, I want to mention that the research and development component of our institute has a science division and engineering division, and also a center who manage the sea operation. So LEOS is only part of it. And so we have a very strong engineering section, as you can tell here. And so it also enables us to do science in this deep sea environment. So our institute, I think, is very unique because of the engineering, the tight connection between the science and engineering. Because as we explore beyond Earth, we also require a huge dependence on engineers. So here just show some of the equipments or instruments that are available in our institute for doing high pressure hydrothermal related topics using Raman detection. We also have a professor who make the diamond and cell also. And in our lab, this is going to be ready in a month, I hope. It's a high pressure chemostat system for for growing microorganisms under this possible condition. So here are some examples of the samples retrieved from the deep sea environment. And we have all these samples collected over the past few years in an archive. And stay in tune because our institute would like to open the archives for proposals if you have brilliant ideas to make use of these precious samples. Talking about astrobiology in China, a little bit of time. So to me, I'm new to China myself as well in terms of scientific research. So astrobiology, to my understanding, is a relatively new term, new discipline in China. Currently have a lower public awareness compared to other disciplines. There's no specific funding program yet. Hopefully in the future there would be. And no structured education training programs yet. I have to say that this means long-term study. So currently there's summer school going on a yearly basis by including some of the audience here, they are part of it. So I'm talking about like bachelor degree program, master degree program that would enable the growth of astrobiology in China. Having said some of these negative things to me, because in China, there are lots of communities, for example, extreme microbiology, biology, space engineering, planetary science. I'm not going to name all of this listed here. So what we need to do is to have some of them to walk out of their comfort zone and work together. Then we have to help the growth to promote astrobiology. And also, there's a lot of opportunities lying ahead of us because you can see the China's roadmap for the planetary exploration. So astrobiologists or astrobiology targeted mission, can that be part of it in some point? I hope so. And as I speak, I think Tianwen, the space shuttle going to Mars, is going to launch today. And so maybe I would stop here. I was planning to share some of the personal note as someone who are so close to astrobiology because I have been studying extreme environment, but I consider I was not part of the astrobiology community yet. And so I was planning to talk about some of the challenges I consider. But anyway, the hope is getting into a new field. There's a steep learning curve here, but as you persist, I think you would be more confident in what you are doing because you know what you are doing. Thank you for, I don't have time to acknowledge every single one whom I work with and I learned from, and here are the funding source that supported all the works that I presented, and thank you for your attention. And as I end here, I would take questions from you as you watch the video obtained from the deep sea by our institute. Thank you.
So Maggie, should I be taking the responsibility of reading the questions? Or
Sure.
you
Or let's see.
or you want to just
Let
read
me
that?
quickly get the video. I hope you get a feel that this is the chimney vent and then the lives microphone around the area.
Oh, you're showing a video. Okay.
So I'm going to just quit this part so I can see the chat. All right. So I see a question from Tony. How robust are extremophiles in less extreme conditions? Could repeated evolution result in changes in their preferred environment or even weakening their environment preferences? Okay, one question at a time. How robust are extremophiles in less extreme conditions? Interestingly, not all of the extremophiles are living at their optimal growth rate under the extreme condition. So there are extreme tolerating organisms living in this place. And so some of them have to be living in the extreme condition, but some of them can live just at the lab bench. And some of them actually you can put them in and out of, for example, a pressure vessel. Yeah, they have no problem with that. Could repeated evolution result in changes in their preferred environment? I am not sure. Changes in the environment must stimulate their adaptive feature. And if they can adapt, I think slowly their genetic material would measure up, would also evolve. And whether repeated evolution, I assume if you consider it's the hot, cold, or wet dry cycles that causes their evolution. In that case, I would say that the only thing I can say that yes, they would evolve. I'm not sure whether it would actually weaken their environmental preferences. But for the organisms, it depends on also the cycle of change, how quickly. So if it changed very quickly, then if the organisms can survive, then I would say that they do have a very wide physiological ranges. But if actually the change is over millions of years, then I'm not sure whether we would still consider them to have a wide physiological behavior. Andre, question. You provided an example from one deep site. How would differences in local affect the syntrophic partnerships that you mentioned? That's a very good question. So the paper was published, so a few years already. As that was the first time talks about the syntrophic partnership in the deep subsurface, but syntrophic partnerships has been known in other places. As far as I know, when they form a syntrophic partnership, it is actually subjected to the environmental condition. And so if the environmental condition allow one of them, favor one of them, then that means they lose the dependence. They don't have to live with the other organisms. And you mentioned about one site, and actually it's the site-to-site variation. Sometimes even the geochemistry may not be that much different. We also see differences in the microbial community. So the factors determining the composition and also their syntrophic partnerships still await for more investigation. Interesting. High pressure chemostat. How much focus do you place on isolation, cultivation of new microbes? Okay. So currently I do not aim for isolation because it's really difficult. I know, Andre, you're more an expert on that. So I'm more interested in enrichment than a single culture in terms of isolating, because of also my awareness of syntrophic partnership. Um, I have intuition that oftentimes when maybe the organisms cannot be cultured in their organisms, partly because they rely on their partner. So the lab condition have to provide a condition that allow both of them to be able to live, not just one of them. And then we don't know much about the nuance between the syntrophic partnership. And so that's why I always more open to doing enrichment to allow us to discover more syntrophic partners. And then the next question, the methanogen to SRM, self-reducing microorganism sequence of partnership. Do these microbes follow same evolutionary sequence of origin? Let me try to understand the question. The methanogen to sulfate reducing organism sequence of partnership. Do these microbes follow same evolutionary sequence of origin? So if, so the person who asked the question, if you could unmute and try to give me a little bit more context, that would be more helpful, because I don't want to answer the question which is actually not really answering your question. Do you want to unmute now, or should I go down more? Let me see if the person is still around. Okay, while waiting, I would answer Iran's question. I have a question about the minimal energy rates you presented in your deep subsurface work. How were the rates for different red dots process calculated for models? Okay, that's a great question. So that's a part of work I learned from work with the TC, my advisor at Princeton. So there's a software called GeoChemist Workbench. And so you plug in red dots processes, half equations. So based on the, not only just the equation, there's a standard Gibbs free energy for each of the equation, and then given the measured concentration of each of the chemicals, we also plug that in to get the free energy flux, because the free energy, the Gibbs free energy, only indicates the energy potential, but the kinetics was not part of it. And so given the chemical concentration will also enable us to tell the likelihood of the energy harnessed by the organisms that use that bioreactions. Okay, and then Maria, thanks for the lecture. My question is, if you are working in polyextremophiles, is considering that in deep sea? Yeah, okay. So I am interested in the organisms living in the in situ condition. So a lot of work I presented today, and they're ongoing in my lab. A lot of them are organisms from the environment, and we characterize the DNA, RNA extracted from the sample. And so I would say that the organisms are subjected to multiple extreme conditions. And so I would say that I have been working on polyextremophiles. But having said that, I need to be careful, because if we do a lab cultivation study, we can subject them under a more defined condition. For example, a combined high pressure, high temperature, and not just one single factor. So that part of work I haven't done yet. Some of the ongoing work would be leaning towards this direction. So I won't read out Andre's answer to Tony. And then in Colombia, we are basically in the same starting points that China in terms of researching and awareness about astrobiology. It would be great if we work together in near future projects. Yes, certainly. I think although this is called ASAP, I think we are trying to be inclusive as possible. And so countries that are less developed in this discipline, even developed country like US, in Europe, I think we should work together because as all the minds come together, I think it would sparkle more interesting questions and ways to address questions. And no problem. Please don't say sorry about the microphone. And please feel free to shoot me an email so I understand better your the context of your question, and I'll try to address as best I could. So Ram has two questions. First is interested to know about the key functional role of bacteria on silica mobility at thermal and non-thermal. I can't really answer that question first because I have only done that one study to understand the silica deposition. And then the one thing that I can say is the silica actually, the structure of silica, there are different types of silica. So under different thermal condition and pressure condition, the structure would be different. And so then how these different silica may affect the growth or have anything to do with the bacteria, that I don't really know. Second, which active bacterial groups other than major methanogens were actively involved in C6-4 flux and had more affiliation with marker genes? Let's see, which active bacterial. I guess this question is more on the permafrost project. So if that's the case, the methanogens are mainly active only in the deep, for example, below the water table, around 50 to 60 centimeter or below. And that's the production of methanogens. And in that permafrost, we have not seen much activity by ANME, the anaerobic methanotroph. And if, or maybe you're asking about fermentation, because fermentation process also produce methane. That may be present in the permafrost, but in the deep subsurface environment, it's not likely because of the very limited carbon, organic carbon in the natural fluid sample.
Maggie.
Yeah.
Okay. I have a couple questions, but before I ask the questions, I want to. You did a really great job with the talk. I think you captured the excitement of the field, astrobiology, very well. But also, I think you realized the challenges that bring people from different fields together, and I think, but you're well positioned to take on the challenge and bring astrobiology to China. So I wish you good luck, and look like the shop is coming up very well. So things seems to be going very well for you. So so the question I have a couple of questions that are very technical. That's all I want to speak. So first question is about the paper that you are involved, that was published in Nature Communications about crucidiopsis. So I actually read the paper. So your conclusion is that the different lineages that occupy different deserts, cold and hot, around the world, the ancestor. There's a common ancestor that was lived on the supercontinent, the Pangaea,
Mm-hmm.
When Pangea broke up, they went on their different ways.
Mm-hmm.
So here's the question then. The breakup occurred a long time ago, like 170, 200 million years ago.
Mm-hmm.
So each lineage would be living on a different continent, in different deserts.
Mm-hmm.
But the deserts are much younger. So the house is young. So for example, in the Mojave, I think you did include a sample from Mojave
Mm-hmm.
or Utah.
Mm-hmm.
The Mojave Desert is only 12,000 years old.
Mm-hmm.
It didn't become a desert, it was a swamp
Mm-hmm.
before that.
Mm-hmm.
It was a very young desert.
Mm-hmm.
So you have a very young home,
Right.
in which you have a very old species,
Right.
very many orders of magnitude older. How could that happen? So that's question number one. So question number two is your observation about the Q10 relationship of methane flux in permafrost,
Right.
where it's linear, which is from 0 to 5, 6 degrees.
Right.
From there up,
Right.
flat.
Right.
The first relationship is understandable.
Mm-hmm.
Chemical reactions increase with temperature. There's always
Mm-hmm.
a Q10 relationship with biological reactions.
Right.
But why does it stop going up
Right.
after 5, 6?
Uh-huh.
So I will stop and hear your answers.
Okay. So very good question. Some of them I haven't really thought so deeply. So I try my best to answer or exchange my thoughts at the spot here. So about the first question, yeah, I think it's a very good way to describe it. The young home with the old lineage. So currently, what we see is the Crucopsidiopsis. Basically, we can find is in the rocks. And so, would they lived? Would rocks be the more recent habitat they have like yield to? I think I see that maybe one of the possibility. So in other words, that means they used to be living maybe in the soil, maybe, or some other habitat. But as the global environment changes, then they all resolve into rocks. So although the desert is young, but their immediate home is not the desert, is the rock. And so our study pay attention on the Crucopsidiopsis that lives in rocks in the desert. I haven't actually looked carefully in the literature. Crucopsidiopsis maybe also happen in the rock in your backyard. What I'm trying to say is that the home, that desert is young, but they have been living in rocks for a long time. And maybe you're trying to get to is because we describe the home hot desert and cold desert, and so that's why I think that gets you into the home situation. I'm not sure I really get a good answer to your question, but that's what I can think of at this moment. Yeah. And then the second question about the methane flux. Yes, we also found it interesting why the Q10 in the early part of the temperature, the thermal range, and then it flattened out. But it is not the first time we see it. So some other, so on the plot, I also put down some other literature. They also see that the methane oxidation takes place at a warmer temperature, but then I would say that it's the physiology of the organisms that we are not well understood. I think that they use the methane. These organisms use the methane as an energy source. But one thing that we can't be sure is this the only carbon source for them? Is methane the only carbon and energy source for them? I always explain to my undergrad mentees when they work on the thesis project is I describe that these organisms are very interesting because they snack on the methane. We don't know whether the methane is their main food, but it definitely has this behavior, and we go after this behavior. Even when we look at the genome, we published the genome early this year. It indicates that it actually has the ability to, it potentially has the ability to use acetate. And few years back, there's a stable isotope study. They actually use 13C acetate, and the 13C actually get into the DNA of this organism. And so it's possible that there's a physiological switch as it gets to a certain temperature. Maybe the energy coming from methane oxidation is not as favorable, or they get more energy by other source. That's one thing. And then because the organisms that we're looking at, it lives in a permafrost environment. So maybe they need to have a quick start as soon as it thaws. Then they need to have like a ramp up the engine. And then they tend to over the few years, what we see is the concentration of the biomass change of these organisms remain very steady. Even the long-term experiment, the thaw experiment, we actually thaw for 18 months. And so these organisms doesn't seem to grow. So that's another thing. I hope one day I would be able to isolate this organism. That would be fantastic. But these organisms have not been isolated. The closest one has been claimed is isolated. I still have stopped about whether this is actually the USC alpha, but it's very closely related. So I would say that the physiology of these organisms would help better answer your question. All right, someone give us a report. Tian-1, the Mars launch is going well so far. Oh, it has been 6 minutes. I'm so sorry I have taken you guys out of this fantastic moment. And I hope that you would be able to watch the news later on. Yeah. So it seems there's no more questions.
Yeah, sounds like that's the end of the questions.
Yeah,
Well,
so
thank you, Maggie, for a fantastic talk. And so I think maybe we can call it the end.
right. So if you guys have more questions, you can shoot me an email. I just type into the chat box. And I also thank you for your compliment. I would say that I was given this opportunity because the Laboratory of Extraterrestrial Ocean system, LEOS, was there before I came. It was the idea of our director, Professor Ding Kang. And so he has been working on deep sea, and to him, the relationship with deep sea and extraterrestrial ocean has a strong link. And so I was fortunate that the place is, yeah, the home is older than than me. Yeah. Okay, thank you everyone, and especially those in the States staying up late and in the other part of the world. Thank you, and I hope in someday we'll see you in person in conferences, or we start connecting through email and maybe even work in science projects.
All right. Thank you, Maggie.
Bye.
Bye.
Good night and have a good day.
Good night. Have a good day.
So I'm the host, so I'm going to close the chat.
Yeah.
Good to see you, Henry.
So Maggie,
Yeah.
since you're not here, so since you will be now focusing on deep ocean studies, deep sea studies, it'll be interesting that you will for a future talk to make a link to the icy world, to the icy oceans, to
Yeah.
the what I call the ice covered oceans.
Yeah. So
really
I
want to hear your take on that.
right. And so as I mentioned, there's challenge getting into astrobiology. So I'm taking my pace to get into the deep sea research as well as the icy world research. And so we have some projects that have relevance. And I hope that in the future years, the relation will be stronger and stronger. And as you said, I really hope one day, and not too long, I will be able to give a talk, including the example from the deep sea environment.
So when I was with Ken Nielsen, there was a lot of discussions in the group about the redox reactions on Europa, because at that time, when astrobiology started at NASA, Europa was really one of the places. One of the things, I think he published a paper with a student, with a postdoc in Science, was talking about, but he didn't say this in the paper. But he realized there was a problem that on Earth, you have unlimited reductant or source reduction or reductant chemicals coming from the vent.
Yeah.
But the oxidants ultimately come from oxygen.
Right.
Oxygen gives you sulfate, give you
Right.
Iron oxide.
Right.
But in the ice-covered ocean, you will not have a supply of oxygen or oxidant.
Right.
Eventually, your redox couples will grind to a halt.
Right.
And I haven't yet heard anybody talk about it. Like, how do you get out of that trap? But I heard the question, heard a rebuttal from what's his name, Bill Schaff. Well, he said on early Earth there was no oxygen. There was the ocean was reduced, everything was reduced. Life persisted fine. So those are really great questions. Recently, I have never heard anybody talk about it.
So I just put up on my screen if you're interested in this paper. Like Kevin's basically in almost every paper on icy moons. Yeah, fantastic guy. So they proposed a paper with an idea that where the oxidants may come from. So it's through radiation that reacts with some organics or that may generate organics. Actually, there are a few papers published in the past 5 years that indicate high energy particles hitting minerals would generate organics. And also, the bombardment onto ice could also generate hydrogen peroxide or oxygen radical particle. And then there are cracks, and the ice, so the oxidants so the proposal here, the hypothesis is that oxidants may form here on the surface, and they may actually flow down, or there's some ice movement. I don't remember what's the time scale, maybe million years. Yeah, okay, don't take that now. So anyway, the oxidants may actually get to here. And so the place where the oxidants may meet the reductants from the deep subsurface would be where the biosphere may occur. And so they propose that somewhere here there may be biofilm. And so the recent mission they detected the plumes on Enceladus, not Europa. They have data to show carbon, more than 200 carbon, like high molecular, relatively higher molecular weight carbon was observed in the plume coming out of the Enceladus. And so one of the proposal would be the plume may pierce through biofilms and bring that brought it into the space. Yeah, so that's the current hypothesis.
Great. No, I have a copy of the paper. I didn't read it yet. So that's
Yeah,
great.
yeah. But that's a great perspective that we need to always bear in mind about the redox availability for the for life to exist. Yeah. All right.
All right.
Thank you so much. Thank you so
you.
much, everyone. And
All right.
yeah. And look forward to Henry's talk next week.